cluster together, it's because you had a match between antibodies and antigens. And so if, say, you had blood and it was type A blood, and you put in the anti-A antigens, then here, these defensive proteins, the anti-A antibodies, will stick to those A antigens and start clustering the red blood cells together. That's what would happen in your bloodstream if they put the wrong blood in you. It would start clogging up every capillary in your body all at once. It'd be like having a heart attack through your entire body all at the same time. And people have died from that in surgical settings when hospitals have messed it up. You hear about people dying from medical errors. There's an example. So I want to make sure if you're in medicine, you know that you do things right. Now, here's a different kind of thing involving complexity of genes. Two different genes contributing to an overall output. That's interaction between genes, one of which has an epistatic effect. One of the genes actually suppresses the effect of another one, producing this range of colors that we see here. What are all these dogs? They're all labs. So we've got black lab, probably most typical, gold lab here, or yellow lab, and a chocolate brown lab. So what happens is, in this case, you have two genes involved with the pigment or the color of the hair, but one of them is controlling whether you're getting either yellow or brown, brown or black pigment. And then another gene is controlling whether it's actually placed into the hair. So what will happen if you have an epistatic effect from gene E, it may prevent any pigment from getting in, and then you just have the yellow dog. That's basically the color of the hair without any pigment added to it. Basically a blonde dog at that point. So you've got these three different outcomes with dogs. So what happens with gene B, you have a big B version which is dominant, and that will encode for black pigment. The only way to get a brown dog is if you have two recessive gene Bs. Then you're making the brown pigment instead of the black pigment. But gene E is controlling whether that pigment gets into the hair. And if you have one or both dominant gene E's, then yes, the pigment gets in the hair. But if it's two little E's, then it's not put into the hair. What color dog do you get in this last case? Black, brown, or yellow? Yellow. Any dog with the little e will be yellow, and it won't matter what this one was having to say. So you end up where you've got four different versions that have at least one dominant or two dominants for both of the genes. And in that case, those are all different ways to make a black dog. It's making the black pigment and it's placing it in the hair. The brown pigment happens if we've got little b, but some capital E, one or both. And then yellow happens anytime it's little e. Doesn't matter what gene B was doing. So you could make this by doing a dihybrid cross, starting with a dog that is totally dominant black, big, and a dog that's totally recessive yellow. You make those together, you produce a dihybrid dog. Looks exactly like the black lab father there. But if we then go and mate one of these to another one, this one and its mates are all producing gametes that have this random assortment of B's and E's. Big, little, big, little. And its mate is doing the same thing over here, producing then this 4x4 Punnett square, but it's a little bit different than the one we saw before. You still get 9 with the dominant phenotypes, where it's, in this case, black dog. But then we end up having 3 of the brown dogs, and then we actually get 4 of the yellows. That's because anything that's little e automatically is a yellow dog. So this is slightly different than the dihybrid cross we saw earlier, even though the underlying math ends up working out the same. So genes interact, producing complex effects. And genes interact with the environment. This is a Himalayan rabbit, so a variety of rabbit that we've bred. And what happens with those is that in the winter, when they're getting their winter coat coming in and temperatures are cold, they end up getting black fur out toward the extremities. There's also a black tail that you can't see here. If this is living in a cold, snowy environment, what would the temperature be like in these areas compared to this area?
Warmer.
There? Warmer or colder?
Warmer.
Actually colder. Think about yourself if you're out in the snow romping around, your nose probably gets cold, your ears probably get cold, unless you have a cap. Your fingertips get cold, the core temperature stays warm. This is the last part to get cold. You go in the extremities, they get cold first. And so in this case, cold activates a gene that makes black pigment. And so as the rabbit is experiencing the winter temperatures coming on, it's developing its winter coat, and this breed of rabbit develops black pigment in the cold spots. Well, a scientist did an experiment with shaving the hair off the back of one and then letting it grow new hair, but when it was growing new hair, he kept a cold pack strapped on. This is a rabbit that was in a captive population, just going around in a yard, in a fenced area. And day after day, the scientist was changing out the cold pack. That's what happened when the new hair came in. So the rabbit ended up with this black patch where it normally would be white, just because of keeping this area a little colder artificially. Didn't hurt the rabbit. It just looked kind of funny for a while. Yeah.
So during the summer, would it be all white?
Probably brown. If you're thinking in terms of camouflage, this is something I wanted to actually find out more about this experiment. Looking into it in the textbooks and online, I just kept finding references saying a scientist did this. I'd like to find the actual paper and look it up. And I was trying to find photos of this. These, I think, are animation. This looks like a real photo, but I don't think those are. So I would like to find actual photos of it, and I haven't been able to. But I think the idea is that the rabbit is camouflaged in the summer, and in this case, it would be camouflaged in winter, except we bred them to have these interesting black ears. But what we did was just fool the genes and cause an unusual development there over a few weeks' time. Genes respond to their environment. That's the point. So let me ask you in that case, imagine that, say, both of your parents got diabetes by age 40. Does that mean you are probably predisposed to get diabetes yourself? And you have a high chance of developing diabetes? It does. It does. Does it mean you must develop it at the same age that they did? No. Are there things we can do to try to prevent the onset of diabetes? Like what? Diet and exercise. Just all the boring stuff they tell us about. Keep your weight down, exercise, try to not eat lots of sugary foods and so forth. And it might be that your parents got it at age 40, but you don't until you're 65. Because genes do respond somewhat to environmental cues. As a result, I like to think about genes not being our destiny. You don't want to just say, well, I got the gene for this, and so there's no hope. There are, in many cases, things you can do, and diabetes is an obvious example. We know very well there are lifestyle things that can help with that. So this is just an image of that about genes responding to the environment. You can change the environment inside that your cells are encountering and lead to healthier outcomes. Now, we have various properties, various traits that are caused by many genes acting together. Again, this is not just the simple on-off binary thing like we saw with Mendel's peas, with, say, the color of the flower. A lot of things in us actually involve many genes interacting. So what happens is that you've got inputs from many different genes combined with inputs from the environment, leading to more of a smooth distribution of things like skin color, for instance, in people. So we get these quantitative traits involving a lot of things that we pay attention to, like a person's height, color of their skin, and so forth. So it's not just one way or another, not just two binary categories. So we've got genes interacting combined with environment, which could be like skin color and the sun, or height could be affected by nutrition when you're growing up, things like that. So we often, with properties that we measure in our populations, we often get a bell curve where there's a lot of individuals that are close to the average and then a few outliers toward the extremes, like this. So this is a common thing to see. You could look at something like musical ability, and if you're looking at people that are playing for the Seattle Symphony, they probably are all right around there. I think I'm over here somewhere, okay? But again, you might imagine that music is involving many different genes interacting. So you've got those genes plus practice, 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 as my sister likes to remind us, because she plays for symphonies. Then those people end up over here, probably based on genes and their upbringing with the training they had and continuous practice. And others of us end up over here where maybe we don't have the aptitude for it and never put in the effort, so that's about where I would be instead. But here's measurements done with height where they actually took some college classes, athletic classes, and so here we had males, here we had females grouped together. And you've got here one very short guy, here's one very tall guy, and then a bunch of guys right around the middle, right around the average, 5'10", 5'11". Here at 5'3", this guy probably got short versions of all the genes that were affecting height. This guy got tall versions of all of those same genes. Same idea here, one outlier, one outlier. So a lot of the properties that are in us are like this, where they're quantitative, involving summations from many genes having their own little inputs. Added together, they produce this kind of thing where there's a lot of ways to be average. Somebody in the middle probably had tall versions of some genes and short versions of others. And the person right above or below maybe had a different mix of those that had added up to the same effect. But there's only one way to get short versions of everything, or tall versions of everything, so you don't get as many of those. And then, that was the idea of one trait, one property, one phenotype coming from many genes. Pleiotropy is standing that idea on its head, one gene affecting many different traits. Sickle cell disease is one of those. That's a disease where you have a lot of different effects that come out, but it's all getting back to a mutation in hemoglobin, changing the shapes of the red blood cells. But you can have symptoms like, for instance, being anemic, obviously, or extreme pain, or organ failure, or paralysis from strokes. There's a lot of different things that go on with sickle cell. And you might, if you didn't know better, you might think these are different diseases, but they're actually all the same disease. They have the same underlying cause. It's a single mutation that leads to this. To get these effects, the person has to be homozygous mutant. They have to have two of these mutant alleles. If they have one normal and one mutant, they're okay. There's another thing like that, cystic fibrosis. And that leads to then again, a mutation to a single protein. In this case, a membrane transporter, and that doesn't work correctly. Should be transporting chloride ions, Cl- ions. Doesn't do that job correctly. And you get, again, a lot of different effects coming out, including pneumonia commonly. Growth is stunted, and the person is often sterile. You might treat those as different diseases, but they're really the same disease. So it's possible for one gene to have many different effects and seemingly produce many different versions of phenotypes. Alternatively, you can have many genes contributing to the same overall effect. And I think I'm going to show you a short film right now dealing with sickle cell. Sickle cell is certainly one of the most serious inherited disorders out there. And it's interesting that it is as common as it is. When you would think that natural selection would want to get rid of it, people with sickle cell disease definitely have a struggle. And yet, it's very common in some populations. And you might think, what's wrong with natural selection? Why isn't it getting rid of this disease? Well, there's an interesting story behind that. So let's see about that story.
Devon and Sky Cooper are brother and sister. Both of them have sickle cell anemia. Before the advent of modern medicine, sickle cell anemia almost certainly meant death before adulthood. Even today, young patients can suffer strokes and organ failure. Sickle cell anemia is a genetic disease. Parents of those who have the disease might not have it themselves, but both must carry the sickle cell character in their DNA.
How's it going? Good. How's your hands? This is pretty. Where'd that come from? Is that from Brooklyn?
Besides some bone pain,
Wow.
Sky leads the fairly normal life of a 13-year-old girl. But her younger brother Devon has suffered acute chest syndrome and has already had his spleen removed.
So you don't get any more belly pains? That's good.
Sky and Devon's symptoms arise from the fact that some of their red blood cells become misshapen, crescents instead of discs, preventing enough oxygen from being delivered to all parts of the body. It's not completely clear why symptoms are variable, but what is most perplexing about sickle cell disease is that it's not rare.
So in the United States, we think there are between 70 and 125,000 persons with sickle cell disease. However, that doesn't take into account immigration and other patients or persons coming from other parts of the world into the country.
In fact, in some populations, African Americans, for example, the incidence is as high as 1 in 500, astoundingly high for a deadly inherited disease. Didn't Darwin teach us that harmful traits disappear from the gene pool through natural selection? Why is sickle cell anemia so prevalent, and why in particular among people of African descent? The answers to these questions began with a remarkable set of observations from an unlikely person more than 60 years ago. Tony Allison has spent most of his career as a medical doctor and molecular biologist in the US and England. But he grew up in East Africa, and he's quick to recall his formative years in Kenya.
The point is that we lived in the upcountry, and we used to go to the coast every year in August for a holiday when it was a little bit cooler than at other times. So we had the trip all the way down, which was usually with a truck and a car. And so we would camp on the way in Tsavo, and there would be lions roaring around, so it was really quite exciting.
These are the infamous Savo lines.
Famous, infamous Tsavo lions.
Around 1950, biologists didn't know a lot about the details of evolution because we didn't know really how heredity worked. The structure of DNA had not been discovered yet. Genetic code had not been cracked. So we know that while evolution was due to genetic changes, we didn't know how the genetic changes took place whatsoever. So there were holes in the whole picture of the evolutionary process. And Tony Allison was probably the least likely person you would imagine who would fill one of the most critical roles. He grew up far away from the centers of science in Europe and North America. he was really interested in natural history, and he loved the Kenyan wildlife, and he visited archaeological digs that were going on at the time. But it was a really circuitous and serendipitous route that led him to an enormous discovery in evolutionary biology.
Tony first went to university in South Africa, where he studied physical anthropology, then to medical school at Oxford. He had a deep interest in human origins, but not so much in ancient stones and bones. Tony was interested in blood. Could the common ABO blood type say anything about the evolutionary history of East African tribal people?
And I actually learned just before going out about this sickle cell condition. And nobody really knew the frequencies of sickle cells in East Africa, so it was a barren slate, so to speak.
Blood samples from people carrying the sickle cell character appear quite normal until oxygen is removed. Tony learned that adding a chemical agent to the samples would quickly reduce oxygen and reveal sickle cells, if they were there. This gave him an easy test to score blood samples for the sickle cell character.
What was striking was that you had high frequencies of people carrying the sickle cell character in the coast and near Lake Victoria, and very low frequencies in the high country in between in Nairobi.
What could possibly account for such a striking disparity? The sickle cell character was understood to be genetic, not environmental. Tony had grown up in the dry Kenyan highlands. He knew the warm, moist lowlands were a breeding ground for the Anopheles mosquito that carried the malaria parasite, Plasmodium falciparum.
And it dawned on him, the places where there was really high incidence of sickle cell was where there was a really high incidence of malaria. Bang.
Now it was a burning question that confronted Tony. Could sickle cell and malaria be connected? And if so, how? It was a radical notion that a genetic disease could somehow be connected to an infection.
When you went back to Oxford, you had this idea of a linkage between sickle cell and malaria, but you hadn't published it. Did you know it was a big deal? I mean, did you?
I was sure it was a big
It was a big deal.
Yes.
So you were.
But that's why I wanted not to go off half-cocked.
Yeah.
I wanted to have a really complete story.
So he decided he had to sit on this idea until he got a chance to test it properly. So and a key element of the scientific method is to come up with a hypothesis. That's great. But you've got to test it in every way possible to see whether or not it can hold up to that sort of scrutiny. That's how science moves forward.
Well, the scientific method essentially means that you address a problem and try to find a solution. And so you look at children of the appropriate age and find out whether they are in fact protected against malaria. And if that's the case, you predict that you will have high frequencies of sickle cells only in areas where malaria is endemic.
You wanted to know that this correlation held not just in Kenya, but everywhere.
It would be important to look directly at the incidence of malaria and sickle cell in as many areas as possible. So, Tony went on a sickle cell safari.
He wanted to gather blood samples from all over East Africa to really test this correlation. And now he was a trained medical doc, so he had something to offer. So he would go into the market on market day and offer to do checkups on children and just take a little finger prick or a little heel prick, get a little sample of blood.
The first thing he did was look at the malaria parasite load in each sample. Then he tested for the sickle cell character. He found that children carrying the character had a lower parasite count, as if they were partially protected against malaria.
And when he examined the blood of about 5,000 individuals, really massive study, the correlation was really clear. So clear, in fact, that he could really draw a map of East Africa and shade in the areas of high incidence of sickle cell, and they were superimposed right on top of the areas of high incidence of malaria. Bang, that was it.
The many samples and detailed maps made it clear there was a connection between sickle cell and malaria. But to understand how sickle cell might protect people from malaria required thinking about the genetics of sickle cell.
What happens is that genes are lined up on the chromosomes, and one has pairs of them with the exception of the sex chromosomes. And this means that you have two copies. So the copies can be the same or they can be different. And if they're the same, they're called homozygous, and if they're different, they're called heterozygous.
When an individual finds a partner and reproduces, one of each pair of chromosomes is passed on. If the parents are both heterozygous, carrying one sickle cell and one normal gene, odds are 1 in 4 that the child will be sickle cell homozygous, 2 in 4 that the child will be heterozygous, and 1 in 4 that the child will carry 2 copies of the normal gene. In the absence of malaria, there is strong selection against the sickle cell gene. However, in a malarial environment, individuals born with 2 copies of the sickle cell gene and those born with 2 copies of the normal gene are both at a disadvantage. One gets sickle cell disease, the other is most vulnerable to malaria. Tony's brilliant insight was that those that carried just one sickle cell gene had an innate resistance to malaria. Malaria tipped the selective balance in favor of heterozygotes. The evolutionary trade-off is that protection from malaria comes at the cost of more sickle cell disease in the population. The sickle cell mutation was not the best genetic solution you might imagine to resist malaria. That's not how evolution works. It was the most available. A simple typo, A to T, in the gene that encodes hemoglobin.
Mistakes are made in the copying of DNA in every generation. You and I were born with about 40 or 50 mutations that didn't exist in either of our parents. It's just part of the nature of copying 3 billion letters in the process of reproduction. And when those mistakes arise, a typo arises in the globin gene. For most of us, that would be a bad thing, but if you live in a malarial area, it gives you an edge against the malarial parasite, so that mutation is retained.
Well, fitness essentially is a measure of whether a particular gene is likely to be passed on to the next generation. And this means that for that to happen, the individual carrying that gene has to survive to reproductive age, and secondly, has to reproduce.
Now you had a sense that you had an explanation that was general to the prevalence of sickle cell and its correlation with malaria. But you didn't quite know the mechanism, right? So
That's right.
what did you do next?
Well, I have to say, I left that part of the story to others because it's quite a complex story. I mean,
A large body of subsequent research has shown that the sickle cell mutation compromises the ability of the parasite to reproduce. Thus, a mutation that creates one genetic disease can also protect against another disease.
What Tony gave us was a fully worked out example of evolution by natural selection. And the amazing thing was this was in humans. This is how natural selection was working on humans in real time, in the real world. Tony's map of East Africa was a stunning achievement, but he could go further than that. He knew that there was a high incidence of sickle cell in southern Europe, in southern India, and in other parts of Africa. And it turns out these were all malarial zones as well. And so his map applied not just to East Africa, but that whole part of the world.
Well, when I'm explaining about the origins of sickle cell disease and its association with malaria to children or their families, they often look at me with incredulity. They don't understand. Like, you're kidding, right? This is all to do with a mosquito infection. As our species has been able to move across the globe to areas with low malarial incidence, this gene is now really more of a nuisance than anything else. There's not really a clear selective advantage for them in Boston, let's say. But it takes thousands of years for the population to change and for genetics to change based on the pressures around them in the environment.
What Tony Allison did, first with his sharp intuition and then with his rigorous research, will stand as a monument, bringing our own evolutionary process into the light.
Now, that disease you know, we get that in the United States. And some people do get sick with it if they inherit 2 of the bad copies of the hemoglobin gene. But we do have a lot of treatments with it, so not as bad as it used to be. I was impressed recently to look at expected life expectancy for a person with sickle cell disease in this country, and it's around 68. That's not too much below the overall life expectancy. Even looking at numbers that were from around 1990, it was much worse. It was more like 48 then. And then, you know, I checked more recently, like I said, it was more like 68 now. So we've gotten quite a bit better at treatments to deal with that. You can even potentially cure it in a person, but to do that, you need to give them the ability to make red blood cells a little differently. You have to do a bone marrow transplant. That's a pretty big deal. But if a person has had a bone marrow transplant, then actually they can be cured of this disease, even though they could still pass it on to their children. So, overall thing with sickle cell is not nearly as bad as it used to be. Without any sort of modern medicine, it would be grim, like you said there at the beginning, where you would likely die from it before you could reproduce. And it does cause a great deal of pain. One guy who was a weatherman, I think back east, who has this condition, I saw a video where he was talking about it. And he said that when you get an attack of that, where you're having an episode, it feels like you're a human pincushion with millions of needles, but they're all on the inside trying to push out your body. That's how he described it. And that's from the blood clogging up capillaries everywhere in sight due to these traffic jams with the irregularly shaped blood cells. Well, all of that condition is something that we'll probably come back to at another time to look at, but otherwise, I'm going to show you some other things with genetics from the next chapter here. Chapter 13. In the first place,